So if we look at our plasma membrane, this is going to be the big part of our discussion now for the rest of the time of this lecture. Of course, I think Dr. Hutchins is going to hack this up into pieces, but anyway, well, for however long we go here, guys, so you'll basically see, because I think we're kind of breaking these up in about 5-10-minute segments for you. But anyway, you'll see the same shirt for a little while, okay, for a few of those segments anyway. So I mentioned the fact we talked about it, a plasma membrane or a liquid membrane. So basically what we have of the cell membrane, and I'll show you this in more detail here in a moment, is we have basically a bunch of lipids, okay, that we kind of refer to as a lipid sea, or to kind of think of it as this expanse of water and its liquidity, and then we have icebergs floating in it. Specifically, these icebergs are proteins. Now, these proteins can basically fit into a couple of categories. They are integral proteins or peripheral proteins. Essentially what that means is that the integral proteins are actually embedded in the lipids, and some of them may even go from one side of the cell to the other, and so we call those transmembranes. And so I think the easiest way to break that down is integral proteins are those that are present in the membrane. Some of them may be peripherally on the inside or the outside, but some of those may be in and go all the way through, and you're going to see a bunch of those today. If I want to move something all the way from one side of the membrane to the other, that protein's got to go all the way through, versus if it only needs to receive information, it may only be on the outside of that cell. But they are in the cell membrane, therefore they are integral proteins. If they're just sitting on one side or the other, then we refer to them specifically as peripheral proteins. And so there's just some terminologies associated with that, and you'll see it. So if we look a little closer at this cell membrane, you can see this. Now, you're introduced to this molecule in the last unit in the biochemistry when we talk about a phospholipid. A phospholipid has a phosphate head and 2 fatty acid tails. And if you look at a segment of our cell membrane, and so notice here's our cell, we've taken a chunk of that and we're showing it here. Notice we have phosphate head, fatty acid tails, phosphate head, fatty acid tails, phosphate head, fatty acid tails. And you also talked about your saturated versus your unsaturated fats and the bent fatty acid chains versus the straight ones. This is what gives that cell its liquidity. Because if you think about when you talked about solids, liquids, and gases, and you said, okay, at room temperature, some of our lipids are solids, and at room temperature, some of them are liquids. Well, the difference being is how much molecular space. So in the case of our saturated fats, we said all of those chains are straight. Therefore, those fats can snuggle really close together. So close cellular space or close molecular space, better said in the context of this, close molecular space, solid at room temperature. You move those a little further apart, liquid. And so if we have a whole bunch of these phospholipids that are bent, they occupy more space. Therefore, they are more liquid in nature. And that's why we have this lipid sea, the sea of lipids. And then we have our proteins. Okay, this would be a transmembrane protein. This would be a transmembrane protein. And we can't tell on these others floating around in this cell membrane. Now, what it creates is it basically creates 2 regions. One of the terms I think you used in the last section was the term amphipathic. Amphipathic being a molecule that has both polar parts, likes water, hydrophilic, and nonpolar parts, hates water, hydrophobic. And so on one side we have the heads, hydrophilic, on the other side we have the tails, hydrophobic. So notice what we've done with this cell membrane is we basically created a phospholipid sandwich where the heads are kind of the bread on the outside, and the fatty acid tails are the meat products, the mayo, lipid mayo, on the inside. And it's because I mentioned that we have water inside, 2/3 of our water is inside, and a third is outside. So we have water outside, water inside. So where do those heads want to be? Well, if that's the hydrophilic region, well, that's where they want to be. If it's hydrophobic, well, they're going to try and hide and orient themselves in such a way that they actually hide from the water. And that's what's happened here. Is we have the heads facing the outside, the hydrophilic, and we have the tails facing the inside, the hydrophobic. We do have in the cell membrane, we do have a few molecules of cholesterol, and that actually makes our cell membrane, gives it some integrity. It makes it a little more solid, less liquid, which is an advantage. We just don't want too much cholesterol because that would make our cell membranes a little too stiff, and so we do run into that a little bit. And so you do see some of those, but you can tell here the phospholipids. Okay, phosphate head, 2 fatty acid tails. Some of them being saturated, okay, so therefore straight. Some of them being monounsaturated or polyunsaturated. Some of them bent. Okay, so you hopefully relate to that construction. And even here in the electron micrograph, you can see this a little bit. A couple things. Remember I said that with our eukaryotic cells, the function is we have a membrane-bound nucleus. Well, it's a phospholipid membrane-bound nucleus. And we have membrane-bound organelles, or the structures within the cell, those little pieces and parts. Well, they're membrane-bound as well. And that's why you're seeing multiple lines here on this electron micrograph. So what we're seeing out here is we're seeing the plasma membrane. And the dark regions are the heads. And then the lighter region in the middle, okay, are the tails. Because these, in this case, these stain darker, okay, and these are lighter. This is going to be one of the organelles we talk about, which has a very similar structure to what our cell membrane does. It's called the sarcoplasmic reticulum, and that's what the SR is. But we're looking specifically at the plasma membrane. And notice they're very close, 80 nanometers apart, so there's not a lot of distance between them. So we can kind of zoom in on this plasma membrane just a little bit. So you can see closer up, you can see, okay, here's my phospholipid, here's my head, here's my tails, here's a little bit of cholesterol in that cell membrane. These would be transmembrane proteins because they go all the way through. But if they're in the membrane, we specifically, the general term for them, so the broad term would be integral proteins. Now notice that they all have different shapes, and it's going to be a big part of our discussion today. That some of, one of them has kind of a coiled appearance, and one has kind of a, I don't know, like a bunch of sticks appearance, and one is kind of a big chunk. Okay? All of these proteins, the reason they look different is because their functions are different. Okay? It goes back to our discussion of anatomy versus physiology. Structure denotes function. Well, these proteins are structured a certain way, therefore they function a certain way. And where do we make those proteins? Well, a big part of our discussion in our next module will be your genetics, and that information that's in the nucleus is what dictates those proteins and how they're formed. And so that will be a big part of our discussion. But now we just want to refer to them as just cell membrane proteins, and we'll talk about some of their specific functions here in just a minute as we go through. So this just kind of goes through and just kind of reinforces the idea of peripheral proteins versus integral proteins versus transmembrane proteins, just another kind of reflection of that.